made that thing non-functional by inserting a mistake, just one, in there, at a key spot within that gene. And then they wait to see how long it takes to evolve back into functionality again. When it does do that, it's able to produce a particular, basically the solution goes cloudy. And they can see, oh, okay, it evolved back. And repair that by evolution. Evolution is a very tricky term, though. It's a very non-rigorous term, unfortunately. So then the experiment went to 2 locations within the gene. And that has been running several years now. And it has not repaired itself in a population of millions of bacteria. And the scientist running that experiment is now suspecting it will never repair itself. So what's going on there? It's similar to what you described, is that random chance can be beneficial, but it can also be damaging. So you've got 2 things happening simultaneously. The gene, most of the gene is good. There's only 2 mistakes in it. So you got these random mistakes being made. Let's say you had a long line of text. There were 2 letters that were 2 words spelled wrong, each with only 1 letter wrong. So let's say let's randomly change the letters in the sentence. Well, the problem with that is that the probability you introduce more spelling mistakes is vastly greater than the probability you get just those 2 fixed. You can actually compute what the probabilities are. And at the end of it all, if you have a very small mistake to fix, you might be lucky enough to get it fixed. But the longer it takes, the more damage is occurring in the other sections of the gene. So you're working against time. And eventually that kills you. Because by the time you fix those 2 mistakes over here, you've got 23 more new mistakes to fix now. You're further behind than you were in the first 2 mistakes we introduced. That's answer number 1. Answer number 2 is most people that I talk to, including scientists, have no clue how many mistakes are possible within the entire evolutionary time frame. Let's say 10 billion years. Let's say 10 billion years, and you can actually compute the numbers. Let's say a population of 10 to the 30th power life forms, replicating once every, let's say, 30 minutes, with a mutation rate, and so on and so forth, a generous mutation rate. It turns out that the upper limit for the number of evolutionary trials in the history of Earth is 10 raised to 42 power. That seems like a big number of trials, but that's not even enough to generate 1 protein. On average, you will need about 10 to 200 trials, roughly. 10 to 200 trials to luck out and get 1 protein. There's not enough time in the history of the Earth. And you can actually expand that to the history of the universe. Turns out it's not going to happen in the history of the universe. So that's where...
Even your first argument is there's got to be a problem for Kuhn's multi-universe theory.
Yeah, there is. It's a lot easier to generate a protein. In the end, it's a lot easier to generate the entire thing in 1 shot than to do it through an evolutionary process where with every mistake, it could be a bad one. It's better to just drop all the letters in the pot, shuffle them around. If you want a protein of, say, 300 residues, to pull out 300, see if it's a 300 and look at it, is that right? You know, throw it back. Then it is to start trying to piece them together. Because in that way, you can actually luck out. You can in 1 fell swoop get a normal protein. But how many samplings would you have to do? The universe isn't old enough. So yeah, given enough time, a lot of things can happen. And given an infinite number of universes, everything that has a probability greater than 0 will happen. But there's problems with that too. Because the probability that you will get a brain appearing instead of an entire universe is vastly higher than any universe. It's known as the Boltzmann brain problem. In fact, reality should be littered with pens and pencils and brains and everything else. Instead of universes, you'll get universes, but you get everything in between. And that's the Boltzmann brain problem, is that everything is probably greater. So, now there's a lot of other problems. There's a couple of major problems. You cannot actually have an infinite set, a counter infinite set of reality. And if we want to learn more about that, Google Hilbert's Hotel. David Hilbert was a mathematician. Okay, another question you have over there.
I'm intrigued by this. Fingerprints of intelligent design are all over physical reality.
Yeah.
And secular scientists are studying that same information. Then why don't they see it?
Well, secular science, actually biology is about 50 years behind when it comes to information theory. I would say the first major study, in 1951, Leon Berlin published virtually the same equation that Zostak published in 2003. 50 years later. Virtually not, the biologists have tended to think of information in terms of computers, language, all that stuff, and just assert that biological information is different. But that's not flying as well anymore, because when you look at the genomes of life, there's only 4 different types of base pairs you can have, and they're in a sequence. As soon as you got that, you've got digital information. And if you got it doing something, it's functional information. And so the next step is, well, what does this do? And when scientists see, most of the scientists are not worried about function, because they don't know what to do with it. Part of my research was to look at what does functional information mean in terms of protein three-dimensional structure. And that paper is under review right now. But what I have found, just in my own research, is extremely useful for protein science in predicting and understanding protein structure. Ultimately, protein structure is encoded by information. And how do we use that? How do we interpret the information to understand the structure? So, most of them are not looking at information. And say most of them have. If you look at the theory of common descent, the Darwinian theory of common descent, most biologists, that's completely irrelevant to them in the day-to-day research that they do. What is relevant is variation within whatever organism they're looking at. They call that variation evolution, so if you ask a scientist, they'll say, oh yeah, I couldn't practice what I do without evolution. What they're talking about is the variation within, say, a virus or a bacteria or a baby. But that variation, as we can see, does not entail that it's unlimited. In fact, orphan genes create a situation where the variation is limited for every organism.
I watched on YouTube a debate with this fellow in Calgary, I forget the guy's name, from Minnesota.
Izzy Myers,
Yeah.
is that my debate?
Yeah.
Yeah.
Pardon me?
Yeah, I know the one you're talking about.
And what I found really interesting was that it seemed like you were talking past each other, that you were on different wavelengths. And listening to this presentation now, I thought, I wonder if, you use that on the one side is science and on the other side is philosophy and religion and theology and all that. And I wonder if there's a bit of a pride issue there, that, and it could happen on both sides, that Christians take an assumptive pride about creation and secular scientists take this assumptive pride that they know better than everybody else about Darwinism. And I just wonder if we should all be a little bit more humble and recognize that no matter who we are, we see what in the mirror.
Well, that would be wonderful if that occurred. I think for those, you probably mostly wouldn't have seen that debate, but in his opening argument, we were discussing the existence of God. And he spent most of his opening argument talking about the Hox gene complex, which I would have loved to continue on, because it's an amazing argument for the existence of God. You realize that when you actually figure out the information required to code for the Hox gene complex. But what he did is he took the Hox gene complex as a given, and once you got that Hox gene complex in operation, you can actually get a lot of variation, a lot of changes, a lot of major structural differences with that in mind. But the problem with taking that sort of an approach in a time debate where you have to do your entire argument in 20 minutes, as I find, I'm sure this was over the head of half the people here tonight, and in 20 minutes there's just not enough time to lay the technical background knowledge. So what we have to do is talk about simpler things. That's not really your point. The point is that being humble made those up. We were the ones who said, well, okay, if it can't breed with this thing over here, if this type here can't breed with that type over there, they look sort of the same, so you put them in the same group, but they can't breed, can't reproduce between the two of them. We'll say those are two separate species. Our definitions are just been, we made those up. We need to have a more objective way of determining what these kinds or models of life are. And that's why I'm suggesting here that the orphan genes will tell us. Because if we see the same, we can classify things according to whether or not they have the same orphan genes. So what would be wonderful is if we could start getting more, we do have some DNA results from Neanderthal man, for example. It'd be nice to get a lot more. I've read different things, like about the Neanderthal man, the variation we have within there is less than